Hello and welcome to this video looking at mutations. We're going to be looking at two main different types of mutations, looking at gene mutations and chromosome mutations. Mutations in general are just a change to the genetic material of an organism, so it could be a change to the nucleotide bases of the DNA. So for a gene mutation, it's a permanent alteration in the DNA sequence, which is similar to what I've said there, so DNA sequence. But a chromosome mutation is any change or error that occurs within the chromosome. So we have mutations can either be harmful, advantageous, or they can be neutral. So a harmful mutation could be, for example, it could be a missense mutation, which is an example of a point mutation. We'll go on to that, but it could cause sickle cell anemia, for example, so that's disadvantageous because we've got a disease from it. It could be advantageous, so it could be a mutation that allows humans to digest lactose, to have more nutrients in the diet. Or it could be neutral, so like whether your earlobes are attached or not. So it's just remember that they're not always bad, they could be good, bad, or neutral. If we're going to look at gene mutations first, then we have two different types of gene mutation. We have point mutations, and then we have indel mutations. And point mutations, there are three different types that I'm gonna look at. We have silent mutations, missense mutations, and nonsense mutations, and these are all point mutations. So a silent mutation is a point mutation involving a change to the base triplet, where the triplet still codes for the same amino acid, so the primary structure of the protein, secondary and tertiary structure isn't altered, so there's not really much effect. That's why it's called silent. So for an example, if we had the triplet GAA, it would change to GAG. Now, both of these triplets both code for this amino acid, so it's going to have the same function. There's no effect, and there's certainly no diseases that come from that. A missense mutation is a change to the base triplet sequence that leads to a change in the amino acid sequence, or a different protein. The alteration to the primary structure leads to a change in the tertiary structure, so it changes its shape and prevents it from carrying out its functions. So if we had GAA, which as we've seen is GLU, and then that's the amino acid, but it's changed to GAC. In fact, I did that right, didn't I? GAC. This now is this amino acid. So this is change in the shape and this can result in sickle cell anemia. Where your haemoglobin is a different shape and can't carry out its function. Then we've got nonsense mutations, which is an example of a point mutation which alters the base triplet so it becomes a stop codon. So this is disruptive in truncated because it results in a truncated protein, which is just a protein that's shorter than it should be, so it won't function. So, for example, if we had GAA, which is this amino acid, we would then have, for example, we could have that changed to TAA, which is a stop codon. So this is a truncated protein, and it will be no function, it won't have a function. So this can cause the disease Dutch muscular dystrophy. Okay, so they're the 3 types of point mutations that we're going to look at. I'll just rub these out so we've got some space to then have a look at the indel mutations. Okay, so we're looking at indel mutations, and these ones are 2 types. We've got insertion and deletion, and these are pretty straightforward. So if we had the base sequence, for example, T A C T G, we would now have, we might have C inserted here. So we can see that a frame shift is occurring in that direction. So if nucleotide base pairs are inserted or deleted, because the code is non-overlapping and read in groups of 3 bases or codons, all the subsequent triplets are altered, and that's what a frame shift is. So I'll show you what I mean. So originally this would have coded for amino acid, and so would this, but now this will code for amino acid, and so will this, so they're very different. So they're all going to be different now from onwards. And the same for a deletion. So if we had this same, I'm going to use this same code, we could have T A C T G. So we've got rid of 1A. Now this is going to be the one code, and then whatever be next there as well. Okay, so the frame shift is another frame shift has occurred. When the mRNA from such a mutated gene is translated, the amino acid sequence is disrupted and the secondary and tertiary structure is altered, so the protein cannot carry out its normal function. So okay, so that is our gene mutations. If we can have a look then at chromosomal mutations, there are few different types of these. We have got, I'll make sure we've got room. Gonna start with duplication. And this would be, say if we have a chromosome and there's this region here is duplicated, we would have like that twice in the next chromosome. So a piece of a chromosome may be duplicated, so it's an overexpression of a gene can be harmful because too many of certain proteins or gene regulating nucleic acids may disrupt metabolism, so overexpression. So that might be, if for Down syndrome, like an extra chromosome 21, so that's a duplication probably. So then we also have inversion, which would be, I'm going to use these in different colours. So if we had this part, and there was a chromosome here that we had, these parts would they would swap over. So this is when a section of a chromosome may break off, turn 180 degrees and then join again. Although all the genes are still present, some may be too far away from the regulatory nucleotide sequences to be properly expressed. And what I mean by that is, if we had this nucleotide base here is now here and the regulatory gene is here, then there's too big of a distance for it to work on and to be expressed. Another type of this mutation, again, we do have deletion mutations for chromosomes as well. This is quite simple, so it'd just be that this section here is deleted, and then they're just lost, so that will obviously affect expression. We've got translocation, where a piece of the chromosome breaks off and then becomes attached to another chromosome. So this may also interfere with regulation of the gene and the translated chromosome. So this could break off here and join there, or whatever it might be. And then we have one final one, which is non-disjunction. And this is where one pair of chromosomes they fail to separate, leaving one gamete with an extra chromosome. When fertilised by a normal haploid gamete, the resulting zygote has one extra chromosome, and this is where Down syndrome comes from. So if they failed to separate for chromosome 21, there would be an extra one. Okay. So non-disjunction is a failure to separate. during meiosis. A couple more things to mention about mutations is how they are caused. All kinds of mutation can be caused by like radiation, like UV light. They can be caused by different chemicals. They can be completely random as well. And that is everything to go through for mutations. So thank you for watching.